Assalamualaikum and good morning class. Today you'll be learning about chromosomal mutation. In our previous lesson, you have learned about gene mutation. Okay, so today about chromosomal mutation. Okay, now gene mutation is a small scale mutation, and this chromosomal mutation, chromosome, it has many genes. So chromosomal mutation is a large scale mutation, and under chromosomal mutation there are two types here. Okay, so we go the one, okay, only one out of the two types first. All right, you watch this video first before we proceed any further.
Human beings are made up of millions of cells. Inside every cell lies a nucleus which holds 46 chromosomes. Our chromosomes carry genes which make us into human beings. Our chromosomes are numbered from chromosome 1 to 22, with chromosome 1 being the largest chromosome and chromosome 22 being the smallest. The last pair of chromosomes are the sex chromosomes, with boys having an X and a Y, and girls having two X chromosomes. Chromosomes have a top and a tail, known as the telomeres, and a centerpiece, which is known as the centromere. A paracentric inversion describes a situation where a chromosome has broken in two places. The intervening segment does not contain the centromere, and the segment has turned 180 degrees and reinserted itself upside down. Most people who carry paracentric inversions are healthy. Families and family members are identified with paracentric inversions because of investigations following a family history of recurrent miscarriage, infertility. In order to become a parent, you need to make a mature egg or sperm. Each parent only hands on half their chromosomes into the egg or sperm. Normally, each pair of chromosomes come together, exchange genetic material, known as crossing over, and divide. Problems can arise for paracentric inversion carriers during pairing up, as there is a segment of the chromosome upside down. When a paracentric inversion is present, a loop has to form in one of the chromosomes, so that the alike chromosomal segments and the genes that lie within are all aligned correctly. If a crossover occurs within the loop, two chromosomal segments are produced that are very unbalanced, one with two centromeres, and the other alternative is a chromosome with no centromere. If a baby is conceived with either of these two chromosome products, the baby will miscarry. If crossing over occurs outside the loop, then the chromosomal products will be balanced and the baby will be healthy.
Okay class, so this video apparently is more on about the inversion of chromosomal mutation, okay, which I will further explain to you later. Right, now chromosomal mutation, it is the changes in the number of chromosomes per cell and the changes in the gross structure of a chromosome. So there are two things here. The number of chromosomes changed, okay, and the gross structure, struktur bentuk kromosom itu pun berubah. Okay, so there are two parts of chromosomal mutation here. So for today, we only cover the gross structure of a chromosome. Okay, we go one stage by stage because I don't want you to get confused between these two. The same goes for the gene mutation. Okay, so we already cover up gene mutation, so that is the past. Now focus on chromosomal mutation, on the changes in the gross structure of a chromosome. Now class, chromosomal mutation occur at more than one gene locus. If you recall, the gene mutation occurs only on one gene locus, okay, but for chromosomal mutation, more than one. And then this chromosomal mutation observable under the microscope, meaning to say under the microscope you can see, okay, the number of the chromosome, whereas it changed, okay, become less or more, or the gross structure of the chromosome, the structure becomes different, okay. And this chromosomal mutation will cause changes in the organism characteristics, okay. Alright, changes in the gross structure of a chromosome. Now, what is the changes in the gross structure of a chromosome? It is the breakage of a chromosome due to errors in meiosis or damaging agents such as radiation. So now you have a picture here. Okay, a part of the chromosome break, right? Why? Because of the action of mutagens or errors during meiosis. Now, these changes in the growth structure of a chromosome, also known as chromosomal aberration. Okay, aberration means it deviates from the normal way, meaning to say the structure of the chromosome, it looks different from the original chromosome. All right, just imagine like the video just now, a chromosome that has 2 telomeres. Okay, sorry, a chromosome that has 2 centromeres. Can you picture that? Okay, or a chromosome that lack of centromere. Doesn't that give a different growth structure of a chromosome? It does, right? Okay, this produce a variety of genetic disorder, and there are 4 types, okay, for changes in the growth structure of chromosome. Okay, those 4 types are deletion, duplication, inversion, and translocation of chromosome segments. We go one by one. Now, class, before I proceed with deletion of chromosome segment, do not confuse. Okay, do not confuse deletion of a chromosome segment with the deletion of the gene mutation. All right, it's similar, but it's different. Now, deletion of chromosome segment. I said chromosome segment. A segment of the chromosome suddenly disappear. Okay, so this is the loss of a portion of a chromosome. Okay, as you can see in the picture here. Right. Okay, a portion, okay, of the chromosome disappear. Now, these 2 red arrows here represent the action of mutation agent. Okay, now one of mutation agent is UV, right? Other than that is X-ray. Okay, there are many others. So, one or more small deletions may cause frame shift. Do you recall frame shift? But much larger regions of a chromosome may also be lost. So, meaning to say, deletion, it is not necessarily only involve a small segment, but maybe a larger segment, maybe C, D, and E region, right? If too much information is lost, the deletion is usually fatal to the organism. Okay, just imagine the size of the chromosome. Okay, it becomes shorter, so pairing will be quite impossible. Will cause the normal chromosome to form a loop with the mutated chromosome or with the shorter chromosome during pairing of homologous chromosomes in Profess 1. Okay, can you imagine that this is the, you don't have this in your module. I purposely did that because I want you to draw the formation of the loop. Okay, this is the normal this is the mutated chromosome, okay, that lost a segment, which is the segment D, okay, which is already deleted. So what happens? A normal chromosome will have A, B, C, D, E, F, and G segments. So they want to pair, okay, because these 2 chromosomes are homologous. So during pairing, what happens? A segment will pair with the A segment. The same goes for the B, C, okay. But when it comes to the D, dia tidak pasangan dia. Okay, so it forms a loop. This part of the chromosome forms a loop, right? So the E segment pair with the E, F with the F, and so on. Okay, so now, class, normal chromosome form a loop. Okay, so under deletion of a chromosome segment, the normal chromosome will form a loop when it pair with the mutated chromosome. Okay, remember that. Now, one human syndrome that is due to a deletion is cri du chat, okay, which is in French, okay, and when it's translated to English, it's meant cry of the cat, after the noise made by children with this syndrome. Credichet syndrome is caused by a large deletion from the short arm of chromosome 5. If you recall in the video, okay, in the karyotype, we group this chromosome 1, 2, 3 until 22nd. On the 23rd is for the X and Y chromosome. So now on the 5th chromosome, okay, what happened? One of the chromosome, homologous chromosome, has a shorter arm, right? And this Credichet syndrome usually results in early death. Although many affected individuals show a normal lifespan, it has a variety of effects including respiratory problems. Okay, so meaning to say, normally this Credichet syndrome individual, they will result in early death. Okay, they still can live, but shorter lifespan. But some shown normal lifespan, and those with the normal lifespan will be able to pass on this mutated chromosome to the next generation. Okay, class, please take note. Okay, deletion of a chromosome segment, one of the example, okay, is the Credichet. I think it came out in the exam before. Okay, next is duplication of chromosome segment. Okay, now a detached or deleted particular chromosome segment becomes attached as an extra fragment to a homologous chromosome. Okay, so earlier you have learned a part of the chromosome being deleted. Now that deleted will attach to the homologous chromosome. Okay, so the mutated chromosome will form a loop when it pairs with the normal homologous chromosome due to the unequal length. Okay, now purposely didn't put this part in your module, so I want you to draw it. So this is the normal chromosome. Okay, you can see the segment here, A, B, C, D, E, and F. And then this is the other chromosome, duplicated chromosome, okay, which has duplicated segment of C, D, and E. Okay, so what happens here? A will pair with A, B will pair with B, and so on. And then the C, D, and E, okay, C will pair with C. But the D, E, and F, okay, would want to pair with the C, D, E, and F. Now, what about the CDE? Okay, this section. So the CDE, okay, forms loop. Now, this diagram terbalik, okay. So the mutated chromosome, which is this one, okay, forms loop. Okay, by right now, when you draw this loop drawing in your module, please draw, okay, this mutated chromosome, okay. Instead of upward here, you draw it here, okay, lukis di bawah. Okay, now the point here is the mutated chromosome forms the loop right under duplication of chromosome segment. Okay, so apparently this diagram here shows that this segment, okay, being repeated, okay, on the chromosome. Okay, or you can say that under deletion, this B and C segment being deleted and then transferred here. All right. Phenotypic consequences depends upon the location of the breakpoints where the duplication occur. Now, class, this region here, okay, where the segment of the chromosome is being duplicated is known as the breakpoints. Okay, so the phenotypic consequences, it depends the location of the breakpoints. If the duplicated region does not lie within a gene, you know that a gene can translate protein. Okay, now if the region doesn't lie within a gene, there may be no effect. But if the duplication occurs next to the original region, it is termed a tandem duplication. tandem duplications are important in the evolution of families of related genes, such as the globin family. Globin family means proteins that has heme. You know, proteins that has heme, like your red blood cell erythrocyte, the alpha polypeptide and the beta polypeptide. Polypeptide is the protein, and you know the polypeptide chain, it has a heme, right? Such as the globin family that encode the protein hemoglobin. Example is the evolution of the hemoglobin in mammalian phylogeny. Okay, you Google up what is phylogeny, sort of like traits. The alpha and beta globin gene clusters originated from tandem duplications events from a common ancestral globin gene. Now, class, it is believed that our globin gene, okay, it is only we have this ancestral globin gene, one globin gene, but from this one globin gene, it's sort of like it mutated, okay, mutated that caused us to have alpha, beta, and gamma globin gene. Okay, this is a result of the tandem duplication. Now, class, keep in mind tandem duplication is under duplication of the chromosome segment. Right. Inversion of chromosome segment. Okay, now this is a result when a segment of a chromosome is broken into places. Okay, like the ones that you watch, and then reversed 180 degree and put back together. Okay, I think you can imagine that from the video that we have watched earlier. So during phase 1 of meiosis, what happened? Chromosomes will coil and shorten. Now, during this phase, a normal chromosome will form a loop when it pairs with the mutated inversion chromosome, like what was shown in the video earlier. Often results in no demonstrable phenotypic effect in the individual. If you recall in the video, okay, individual who suffer who has this inversion of chromosome segment, normally they don't demonstrate phenotypic effect. Okay, phenotypic effect means something that can be observed. All right, so you can't see that they are having this inversion of chromosome segment from the outside. So you can see from this example here, right? Let me get my pointer. All right, so this segment of the chromosome which is exposed to the mutation, okay, it becomes inverted and then it broken into two places and then inverted and put back together. So you have this outcome, BCD becomes DCB. Okay, so this is the normal chromosomes. It will form a loop in this sequence A, B, C, D, E, F, G, H, I. Okay, because the inverted chromosome, if you look here, right, let me change the in color. Okay, AB becomes G, F, E, D, C. Okay, and H terbalik, sir. So the normal chromosome have to form a loop, okay, so that it can pair, the chromosome segment can pair with the chromosome segment from another homologous chromosome. Okay, now there are two types of chromosome inversion. Okay, one is a paracentric and the other one is a paracentric. So you have this diagram in your module, but you have to label them. Okay, which one is the peri, which one is the paracentric. Now, pericentric is the inversion of the chromosome that includes the centromere. So, I mean to say, the segment of the chromosome, okay, being removed and then reversed. Now, the segment of the chromosome, the thing is, it includes the centromere, as you can see from here, right? And then being put back. In paracentric, okay, inversion of the chromosome, it does not include the centromere. You can see the centromere is here. It doesn't disturb or include the centromere. Are you clear with that? Okay, I hope so. Okay, the last part, translocation of chromosome segment. Okay, you don't have this diagram in your module. Purposely did that because I want you to draw, okay, this chromosome diagram. Now, translocation of chromosome segment is when a chromosome segment detached and reattached to a different location of the same chromosome. Okay, now there are two types of translocation of chromosome segment: internal translocation and cross translocation. Okay, now in internal translocation, okay, it is the transfer of chromosome segment to another section of the same chromosome, okay, which tally to the general definition for this translocation of chromosome segment. All right, remember, a part of the chromosome segment is being detached and reattached, okay, on the same chromosome but on a different location. So internal translocation, transfer of chromosome segment to another section of the same chromosome. All right, so you can see from here, okay, the segment E is being detached, okay, and reattached, but on the same chromosome, only in the different location. Okay, so okay, internal translocation, you refer to this diagram. So you draw this diagram now. Okay, done on your drawing. All right, now the second type of translocation of chromosome segment is the cross translocation. And cross translocation, there are two different categories: non-reciprocal translocation and reciprocal translocation. Cross translocation, okay, cross, silang. So from here, a segment of the chromosome must be transferred to another chromosome. Okay, that's the difference here between internal translocation and cross translocation. Internal hanya melibatkan kromosom yang sama. Cross translocation, a segment is being transferred to another chromosome. Okay, now slightly I understood the definition for cross translocation slightly not tally with the general definition for the translocation of chromosome segment. Now this definition here only define the general subject here. Okay, right now let's get back to cross translocation. Now cross translocation, okay, as I've said, there are two types. So now the non-reciprocal translocation means donation of chromosome segment from a chromosome to another non-homologous chromosome, okay, but receives none in return. So meaning to say, one chromosome, it donates, okay, one of its segment to another non-homologous chromosome. That's it, end of the story. It doesn't receive any return, any chromosome segment in return. That's it. Okay, so this is known as non-reciprocal translocation, and this leads to deletion and duplication. Remember, you have learned this earlier. So deletion means that chromosome, okay, one of the segments is being deleted and received by another non-homologous chromosome. So that non-homologous chromosome that received the deleted segment becomes duplicated. Okay, now another one of cross translocation is the reciprocal translocation, which is the transfer of chromosome segment between two non-homologous chromosomes. Okay, so two non-homologous chromosomes transfer their segment. So one transfer and receive, the other one receive and then transfer, things like that. Okay, the diagram, okay, for the non-reciprocal translocation, I think you can understood, right? Or you can draw on your own. Okay, and the reciprocal translocation diagram is here. Okay, so apparently this segment transfer here, and then this segment is being transferred here. So you get this outcome. right? All right, test your knowledge. One possible result. Okay, again class, you do these questions on your own first. After you have done, then you come back here and cross-check with your answer. One possible result of chromosomal breakage is for a fragment to join a non-homologous chromosome. What is this alteration called? Okay, it's translocation. Okay, a non-reciprocal crossover causes which of the following products? Deletion and duplication. A non-reciprocal crossover. I just mentioned this. Okay, as well as this one. Okay, an inversion in a human chromosome often results in no demonstrable phenotypic effect in the individual. What else may occur? Okay, inversion. Now this is about inversion. Individual who suffer from this inversion or chromosome mutation, they didn't show any phenotypic effect, right? But what happens to their chromosome? Okay, they might have abnormal gametes. Okay, you have watched the video. The gametes might be abnormal, and this will lead to, okay, when they married with someone else, okay, miscarriage, right? Watch the video. In order for chromosomes to undergo inversion or translocation, which of the following is required? Okay, which one? Inversion or translocation. So it must involve a part of the chromosome to be breakage and rejoining. Okay, now class, draw a mind map that signified the 4 different types of changes in the growth structure of a chromosome. So now, this is your major homework. So you have studied the 4 different types of the changes in the growth structure of chromosome. Now I want you to draw a mind map, okay, that relates these 4 changes in the growth structure of a chromosome. Okay, use a mind tree map or any mind map. Okay, all right, I think that's it for today. Okay, thank you. Assalamualaikum.